We're not even interested in cells anymore. We're interested in DNA. DNA is not living, right? So we've passed from the notion of, I mean, in Foucaultian sense, he thinks about, you know, when we move from natural history to biology. It's the beginning of, biology is about the study of life itself, right? We're beyond the study of life itself. If anything, we've grown kind of unconcerned with that, and what we're really fascinated with are these these micromolecules inside of living things. So we've gone back to a kind of, I think we've left biology at this point.
I got a half jug of agar and an old PCR machine. I got a half a jug of agar and an old PCR machine.
One of the more important experiments in terms of self-experimentation was the development of the PCR process, the process of polymerase chain reaction in 1985, credited to primarily Kary Mullis, who later won the Nobel Prize. I think of this as being a really interesting kind of experiments in explorations in identity, because not only does this technology furnish us with a lot of what we know about self-identity via a lot of genetic mapping and genetic imaging, but also Kary Mullis, the founder, was said to have told persons that he had discovered this under the influence of LSD with a fluorescent green raccoon as a sort of spiritual muse. You want to see blue? But glow in the dark fluorescent green raccoon. Is there something you can tell me?
Mama said I could eat a marshmallow.
I'm lucky. I was very lucky to have the fluorescent raccoon here, because the fluorescent raccoon here told me what I needed to know, that while all the others that were doing similar kinds of things with DNA and trying to find things on the genome, they were trying to amplify the signal. They kept trying to figure out how to make DNA glow more brightly and like making better radioactive probes and all these things. But what Mullis realized was that via the fluorescent raccoon was that what you really needed to do was to make more sample. Not more signal, but more sample. So then, rather than try to amplify that signal, he amplified the sample by billions of times. The sort of primary interesting player in the PCR process is a special type of bacteria called Thermus aquaticus. Thermus aquaticus lives in almost all hot springs, and the original strain that was used in these experiments was isolated in Yellowstone National Park. Actually, it's pretty much stolen from the national parks, because national parks never received the billions of dollars that that organism generated. What I'm going to be doing here is to try to find that organism, try to pull out its enzymes that could be used in this TAC process or in this PCR process. Once I have those, I'm going to try to perform PCR, not in a lab, not in a very specialized machine, as most labs would, but I'm actually going to try to perform this over the campfire.
When we're in the bus, just remember to charge the drill battery.
Let's spark this place up.
All right,
Let's see what it's got. See what this lab's made of. So this is it should be what's called LB, Luria broth. Nice stuff for E. coli to eat. And it also turns out that the TAC bacteria from my studies have seemed to thrive on this as well. This is a vitamin stock specifically for Thermus aquaticus, which is the bacteria that we're hoping to isolate here. So we want these things not only to be able to eat, but to be able to breed. And so to do so, we need the right amount of foodstuffs for it to eat, and the right amount of vitamins and minerals for it to, that it also needs to survive. TAC is a pretty interesting bacteria. It's not just your standard deal. It's a long, filamentous thing. It looks like a giant snake of the, or python of the bacteria world. It's got a flagella at the end so it can even move around to make it smell a little bit like the center of the earth. I'm afraid my bacteria is pretty unisex, you know, as far as I know. There's some have written about the fact that it can also be a spore-formulating organism, so it can have a kind of 2-cycle lifespan, but I don't understand that part of it. All I know is what it eats and where it likes to breed in. And I want to get the real classic-looking stuff. I don't want the spores. I want the real filamentous buggers. I always find that the most important thing with all this biology stuff is just to be pretty organized about what things are which. So the Thermus aquaticus bacteria, particularly the polymerase that it creates for copying its own DNA, is thermally stable at these very high temperatures. And so it's that quality that allows us to produce, when we amplify DNA, literally billions of copies. The reason this is relevant to the area is that the hot springs in this area are where this kind of bacteria is native. Originally, they were isolated in Yellowstone National Park, but they've since been found in many of the hot springs around. We're going to see if this one works as well as the kind of classic Yellowstone variety. This is an incubator, and we should be able to set it for whatever temperature we need. We'll probably have to monitor it a little bit, but we want to take this all the way up to 70 degrees Celsius.
For how long?
For a couple days. They like to be kind of shaking around a little bit, because
in the womb or in nature.
kind of, yeah. I mean, basically everything with these things, they need to be able to get air, and so they need to be able to kind of get sometimes recycled to the surface. We'll see. We'll wait to see about the film.
Just wait out and the moose kind of sit there.
Just a matter of waiting. It's still quite early. It hasn't even been.
And the guys go like this.
It's only been about 12 hours of actual heating. So we'll see. We'll see what happens in there. So I can use standard table salt. Here we go. 2.3 should be fine. This kind of disrupts the loosest dead cells in your mouth. Let's just shake this up until nothing's on the bottom. Something just disgusting about holding salt water in my mouth. Damn. Ugh.
They're getting bigger in the lab.
Yeah, Jesus Christ. As long as, I mean, this is a little bit disgusting, but it is only my DNA. It's only my spit after all. Should be pretty similar heights too. So then these are going to go into these are going to go in the centrifuge. I'm assuming full speed is 1400 RPMs. The centrifuge on there's a little bit more fingers in the spit than normal. That's spit stuff, so nobody should drink that.
Okay. When you're done with it, you can put it in the bio-waste container.
We can. This little pellet there, you probably can't see it, but that little pellet at the bottom are the squished up cells. What you kind of do is you flick the little stuff around there or resuspend it into the fluid so that it's not so absolutely compressed. Yeah, good. When the cells burst, it will stick to all the cell membranes, but it won't stick to the DNA. And so that when I finally pull this stuff out of the hot water baths, I should be able to like spin it down in the centrifuge again. All the cell membranes and crap will go to the bottom, and then the DNA will be left in the fluid, and that fluid then will be what we're looking for. Okay. These are my sort of more inexact thermometers. So this one we want to get to 100 degrees, and this one we want to get to 56 degrees. And if this one gets too hot, we'll slow it down with some of that water. Keep stuff rolling.
Oh, yeah.
We got them today.
Make a little mistake there, team. Nothing is ever perfect. When I put the cells in, they're a little clumpier than I wanted them to be, and they sat a little longer before I flicked them around than I wanted. And yeah, it's very hard to find the perfect conditions. I've yet to find those. 56 degrees centigrade water bath. Put them into the 100. What we hope is about 100 centigrade water bath. There we go. We're at 130.
It's perfect.
So 132 degrees Fahrenheit is where we want our.
That's Ian's, that's right.
Between the salt and the between the chemicals and the heat, everything should be lysed open, broken open, and the beads should be, I think holding all the cellular components with the exception of DNA. It should be binding to them, and then when we take it inside, we'll centrifuge it down, and we'll centrifuge it at a speed that's fast enough that all the cellular crud and the beads get stuck to the bottom, but not so fast that the DNA does. And so the DNA will stay in solution. So we'll suck the DNA out of the top of the tubes, throw out the bottom, throw out the stuff at the bottom, and then we'll have 4 tubes of about 100 milliliters of, or 100 microliters of decent usable DNA. Amplify the signal, or the sample, not the signal. You know, that was the basis for everything in the experiment working, right? Usually the new ones all have this conversion between the speed, the spinning speed, and the actual amount of g-forces it generates. But this one doesn't. Sometimes when you work with a lot of older equipment or secondhand surplus equipment that hasn't exactly been calibrated perfectly and whatever, you do wind up kind of doing, you kind of learn how to use those machines, and you realize that there's a big difference, or there's a difference between this idea of like what is exact and what is consistent. But sometimes just being consistent is good enough. For instance, particularly with measurements, I mean, it's provided that you're using something that's off by the same percentages each time, and you're measuring every single ingredient with it, then you know you might be scaling up or scaling down, but you're actually getting the same ratios. So what I want to do is I don't want to scoop that. All that stuff is a leftover cell mass and a bunch of other crap, which we don't want. So, but that's 1 tube of DNA for tomorrow. Enough lens on your camera. You can also do something, one of these. Looking through these puppies, you can simply. These are some of the decent images. These are some of the guys we're looking for. The tack. There's lots of little. I can maybe blow it up a little bit. You can see all those little wormy things. Well, I mean, it's just like any time you do this, there's always things that are imperfect, you know, and this is no different. So I think in this time it seemed imperfect that the sample sat for more than 24 hours after harvesting, and that's not great. And we don't know exactly how far bacteria would have traveled down that spring from where they're actually really thriving. So it could be that they've been traveling for days without proper nutrients anyway. So we don't really know. So the idea here is just to try to get some quick results about if one of these is viable. Remember to swish them around a bit first. If one of these is actually viable, we're gonna say great. We're gonna harvest up the stuff from it, and we're done, right? I mean, methylene blue is kind of a methylene blue is kind of a classic. So I think we're gonna try. Usually 1 drop should be perfect, and hopefully this comes out in a nice dropped form. 1 drop should be about perfect if this is a similar concentration to food coloring, I'm hoping. Particularly with, ooh, that's a dead mosquito, it's not. Then you want to flick it around a lot because if Thermus aquaticus is in there, it tends to form into big matted kind of sheets, and so you want to knock it up enough that yeah, so we can see the individual ones. Drop about like that. And then a piece of cover slide.
So this is what you can with a pre-made.
What we're talking about, Thermus aquaticus, it could be there, and it might have just grown very strangely because of something about the circumstances. I'm just checking to see if anything grew in that solution we made on day 1 of the camp here. I don't think much worked. Not sure why, but I'm going to double check before I say it didn't work. Got it. Just turned out to be the lens needed a good cleaning. Wiggle that between your fingers for a little bit. 2 different sets of PCR reactions we'll do today.
So this is what you made this afternoon? Or?
Yeah.
Okay.
This is everything needed for the PCR reaction. In that, 20 in that, 20 in that. Usually everything's written down in really precise format. Usually I would make an actual log of exactly how much of everything needed to go into each tube. In this case, I've done this enough times, and the level of you know, we're trying to be a little more casual here in the woods. Yeah, so I'm kind of modifying my own recipe on the fly, which is, again, it's not, don't try this at home. This is a lot of tricky math to keep in your head. So I'm kind of trying to do all these sort of in my head right now, just sort of keeping track of the numbers. So I'm hoping we don't get into trouble. So like a lot of times if you're doing a big PCR experiment, you're doing 60 different tubes of stuff, so there's a little bit more potential for error. With this, just doing 8 tubes. What I found with PCRing is these, right now I'm doing a pretty small region of DNA. I'm doing a kind of standard size region. I'm trying to amplify regions of DNA that are about 500 to 600 base pairs long. That's actually the range that's usually pretty easy to do. I gotta put some of this stuff back in the fridge, and then I'm gonna take this stuff out to the fire.
Sure. Okay. So I'm gonna change my tape.
Okay.
So it's a song about a guy who travels around the countryside breeding genetically modified pets. His woman left him, and he's got some other problems too.
We're sorry, Mom.
And I've never played it like this, so we'll see how it goes. Not at all.
Like a Canadian witch.
Yeah, exactly. La 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 la. Okay. We're dogging on the muggy oak trail. We're dogging on the muggy oak trail.
15?
Feel free to like, I know,
23,
like
23,
coyotes,
14, I'm
like whatever you want. You never really loved me, darling. You never did, and that's a fact. We're dogging on the muggy oak trail. I got a half jug of agar and an old PCR machine. I got a half a jug of agar in an old PCR machine. I'm gonna let the acid bubble. I gotta keep that bottle clean. And I never seen so many colors, not even in a dream. I gotta let the acid bubble, gonna keep that bottle clean.
So Casey found a nice little spot down here for electrophoresis. Good a spot as any. This little jar can go here. That's going to be our what we call the tip jar. Yeah, this is the gel we made this afternoon. This is just a very standard agarose gel. And then I need the liquid here, thanks. A buffer solution. This buffer is so that I'm gonna basically make an electrical field here. And the buffer is so that the field is consistent. Yeah, but see, the water looks lower there, looks higher there, right? Than here?
Yeah, it's higher this
Right.
side.
Right?
Yeah.
Yeah, that looks perfect. Okay. I'll get these all set up and ready to roll.
You're good, Paul.
Thanks.
One thing is
Yeah.
Scientists to do art.
Couple of them though, then it was brilliant.
The sun is
Maybe one more of this. I'm gonna put one more of these in there. Song is gorgeous.
yeah.
I'm running a electrophoresis gel, which is the technical term for what is usually called, DNA fingerprinting or genetic imaging or whatever. It's just a way of testing the size of the DNA that you have. And so I'm just testing, by testing the size, how many base pairs are in my DNA samples, I'll determine a little bit about my own identity, and hopefully we'll even be able to determine something about my identity, because we did this PCR over the flames last night of the fire, and if it worked, we will then be able to tell certain things about me, whether I'm heterogeneous or homogeneous for certain kind of traits. Because one of the things I was looking at was the presence or absence of a gene called the ALU gene, and that's like a kind of a standard marker in populations, because about 1/3 of the people are homogeneous for one trait, another third are homogeneous for the other, and the third are heterogeneous, meaning that they have both. And we'll see which one I have. I don't know. And then
Can you speculate? Would you
Yeah.
sort
What
of,
would you like?
I mean,
What would you like?
I always think of myself as being a rather heterogeneous fellow, sort of a mixture, no purity in myself. So I would expect that my DNA is likewise rather impure. I'm a post-race kind of thinker, I think race is a colonial construct, and thus I'm against the whole notion of racialization, so in a way a lot of this work is.
And how is
positioned
that
that way.
working against the notion of race?
Well, in this case, this is a little more playful because I'm doing this kind of more I guess, 19th century kind of romantic notion of the individual. I don't know where it fits in there. There is something essentialist about that 19th century vision, but I'm not. So in a way, I guess I'm complicating it with this kind of work in this specific context.
You're raising the problem rather than bringing the solution.
Yes, precisely. Yes, precisely. And I think of it as a different way. I really think of this as post-biology.
In what sense? Because what I was saying is was coming from Henri Atlan.
Yeah.
He says that biology passed from being a science of observation to a biotechnology because now it creates artifacts.
I fully agree that it passed from being a science of observation. I think it passed from a science of observation to a science of rationalization. And that's why I consider it post-biology, because I think it's more informatic and statistical than it is necessarily biology.
So
just in the same.
is like removing biology from its own.
Yeah, sure. We're not even interested in cells. I mean, we're interested in DNA. DNA is not living, right? So we've passed from the notion of, I mean, in Foucaultian sense, he thinks about, when we move from natural history to biology. It's the beginning of, biology is about the study of life itself, right? We're beyond the study of life itself. If anything, we've grown kind of unconcerned with that, and what we're really fascinated with are these these micromolecules inside of living things. So we've gone back to a kind of, I think we've left biology at this point. In biology, the cell was the basic unit of life, and so it's really the sort of central object of study. Now, in a way, it's kind of a byproduct of informatics, right? I mean, informatics are the basic, are what life is, right? We can strip these. We can strip a cell down to nothingness and then kind of rebuild DNA molecularly, synthetically, and insert it, and thus DNA becomes now the sort of central unit of investigation. My critique is, I mean, I'm critical of this kind of essentialization. I'm critical of the kind of this sort of genetic determinism that comes with this kind of strongly kind of DNA-centric science. But I'm also, I think, just pointing out here that we may have left, what we formerly would described as biology, right? We may be in a different, not that biology doesn't still exist, but the current era of big science is not biology. I think it's post-biology.
So post-bio art creme is more appropriate.
Yes.
So it's happening?
Yeah, it's happening. It's running a little slowly, but I think it's working. Yeah, you can see how, particularly with the samples on the left and the right, how the purple band is moving forward very quickly. Okay. It worked flawlessly. Every single lane.
Wow.
Yeah, we just need to be in the perfect darkness.
Oh, okay.
Yep. Okay. You can come and look.
You go. Just need to have a hand.
So those glowing bands
So Paul.
are the different gene segments that were amplified, and I used very subtle variations in my mixtures. So that's why I'm getting more than one. They're very subtle variations. I was thinking maybe one would work.
And the single one is?
So this basically is just, this was a region of a skin color gene, the SRC gene.
Oh.
If we're getting these kind of really high amounts of DNA
days after that, then
from over the campfire, yeah, I mean, it does kind of call into question the current paradigm.